Hello, hello, hello. Hello, hello, hello. Can't see anybody yet. But hopefully there'll be a few. Let's just try and get that. Oops. 32. Hello, 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 hello. I can't see anybody just yet, and I can't hear you. But I hope you're ready for a whirlwind tour of plants. Let's just see if I can see. Aha! Aha! Law! Hey, Law! Angela Mackenzie! I can see some names. Fantastic. Lily, hi Lily, how you doing? Angela, do let me know when you come on board so I can see where you're from, who you are. Jude, how are we doing? Manya, hello Manya. Lovely to have you on board. Jordan, hello Jordan. Farley, Jonah, Edley. Sawyer, Sadie, Reagan. Hello Reagan, hi Killian. Cool. And Angela from Inverness. Inverness is so far away. Hello, Manu from India. But the weather's a lot better there than it is here at the moment. Fantastic. Hi, JC. And who else have we got? A couple extras. Wait a couple more minutes just for you to sort of load on. Excellent. Becky, hi, this is Hinton from Clay. Hello, Hinton, all the way from USA. Hi, Katie. Hi, hello. I can't hear you. Not sure why you can't hear me. I can't see you and I can't hear you, but I can see the comments occasionally. We're going to go through a period where I'll do the experiments, etc., and explain some of the science and highlighter, and then we'll have a Q&A session right at the end, and then that'll open up. So it sort of saves all the sort of loads and loads of sort of queries and questions and stuff like that nature. Hi, Aaron. Hi, Kieran. Hi, Yuan. So hopefully, without much further ado, I shall crack on. We shall start. So for those of you who haven't seen me before, my name is Dan, and today we are going to look at the MEL Chemistry of Plants. So it's this particular box you actually need. Sometimes the box is a little bit different, but hopefully, this is the one we're going to be looking at today. So this is Chemistry of Plants. Okay. So as you can probably guess, plants is an enormous field. So many chemicals, so much to talk about. So we're going to boil it down to 3 basic chemicals today, and we'll come on board in a second. So without much further ado, we need a quick equipment check. So quick equipment check. So today you need, in no particular order, but I will start with the chemicals. You will need the sodium carbonate. So you need sodium carbonate, you need the potassium iodide, you need the citric acid, and you need the copper sulfate. So 4 chemicals. You need 4 chemicals today. Hi Max. You need sodium carbonate, potassium iodide, citric acid, and copper sulfate. You need your board, your little tiny reaction tray from the starter kit. You also need 3 or 4 little cups from your starter kit. You need the filter funnel from the starter kit. And you also need the conical flask from the starter kit. You also need today, the pre-prep things, you needed some potato, you need a bit of potato, just 1 potato will do. You need some lemon, so 1 lemon. You don't need a banana, but if you wanted to do a banana later, you can do that, that's perfectly fine. You needed some cabbage. So I'll just go back to the screen here. You need some cabbage. Now this is just chopped up cabbage, of course. And then you basically put boiling water in here and then just leave it set for about sort of 10-15 minutes, something like that. And it's the purple cabbage juice that we're actually looking for in that. Okay, what else do we need? We also need some hot water. So this is some boiling hot water in a flask. It'd be handy if you had a knife to sort of chop up some of the fruit. You need from your pack also the litmus paper. So this is some of the litmus paper. You need 2 of the syringes, so 2 syringes, 2 of the spoons. This is the little tri-pack absorbent pad. I've got a little cutting tray here, so if you've got one of those, that's handy, but you don't need it. You can do everything on this tray. You also need 4 of the little test tubes. So this kind of mini test tubes with their little lids on. Why do you need Halla? You need the red cabbage. you need the red cabbage, because that's one of the investigations we're going to do. We're going to look at a group of chemicals called anthocyanins, which you find in red cabbage. Okay. Okay, so what else do we need? What else do we need? You need one piece of filter paper. So this is a piece of filter paper. In your pack, you have this one piece of square paper. You also need your safety specs. You'll need some gloves. And I think, some access to some water would be handy. So some access to some water just to rinse things off a little bit. And then I think we're good to go. I think we're good to go. So I just want to check. Is everybody okay with this? Everybody okay? Shall I stop? I'm going to stop now so when people ask me some questions, if I've gone too fast, then I can see. So if you've got some questions, I will spend a couple of seconds now waiting for the questions. Anybody got some questions? I know I go a little bit fast sometimes. I get a bit carried away, a bit excited. Everybody okay? So no more questions. Okay, cool. Right. So where do we begin? So this chemistry is all about plants. And in particular, we are going to look at 3 chemicals. What can we substitute cabbage? Jordan, you could substitute red jam, you could substitute roses, you could substitute tulips and tulip flowers. You could substitute some beetroot. In fact, got some beetroot that would do. It'll produce a different series of colours, but it'll still work because they still act as indicators. That's what we're going to come on to. Okay, Manuel. So when we look at plants, of course, it's a huge range of plants, and plants are incredibly important, of course. You know, without plants, there'd be no oxygen, no animals, no us, no food. The planet would be barren. So plants are pretty important. And today, obviously, we only have the time to look at 3 specific chemicals. So, and those 3 chemicals, one is anthocyanins. So plants produce anthocyanins. They also, which are sort of the purple colored things, the dark colors in fruits like blueberries and blackberries and stuff like that. Those are called anthocyanins, those chemicals that give that kind of color. We're also going to look at a substance called ascorbic acid, vitamin C, ascorbic acid. So most plants produce ascorbic acid, and so we're going to look at that. And then also all plants produce starch. We're going to look at starch. So we're going to analyze those 3 things, the 3 chemicals. Everybody okay so far? Yeah, so if you've got any more questions, I'm hoping I'm going about the right sort of speed. Anybody, any questions? And of course, I'll throw a couple of jokes in if you're interested in a couple of jokes. I hope they're funny. And then there's a Q&A session at the end, so I can cover any questions that you may or may not have at that point. Okay, if I get a couple of okays, if I get about 3 or 4 okays, jokes, there'll be jokes coming. So we'll do a couple of jokes after each of the experiments. Okay, right, so if I get a couple of okays, I'm going to assume I can crack on. Is everybody okay for me to start? Okay, okay, Daniel, okay. I've got a couple of okays, kind of okay. Alicia, fantastic. Okay, Jake, you're back. Jake, sorry. Good, I'm sorry I've been away for such a long time, but back now. So, and then more regularly from next week as well and so forth. Cool. Right. We're going to start then with the cabbage. So we're going to start with anthocyanins, okay? So I'm going to move all this stuff aside. I'm just going to focus on here and show you what I'm operating with, first of all. So the chemicals you actually need for the anthocyanin, for the red cabbage, you need these 2 chemicals in particular. I'll just hold these up for you. So you need citric acid, so it's an acid, and you need sodium carbonate, which is an alkali, or produces an alkaline solution. So you need these 2 chemicals to start with, okay? So these 2 to start with. You need your pipette. So we're going to move those chemicals off to one side. You also need your little pack of litmus paper. I'll explain what litmus paper is in a second, okay? So you need your little packet of litmus paper. You need your filter funnel, so your filter funnel and your filter, and you also need your filter paper. You need your filter paper, okay? And your red cabbage juice. So you need your red cabbage juice. If you've got rose petals, that's fine. If you've got beetroot, that's perfectly fine. So all these things are okay. So the first little experiment we're going to do is this one here. We're going to do the anthocyanin one, okay? So just to quickly recap on that, anthocyanins are a group of chemicals that plants produce that give it the colours. There's lots of different uses of anthocyanins for plants, not just for colours particularly, but in our case here, we are interested in the fact that these anthocyanins can be used as indicators. So they will change colour. So that word again, indicator, they will change colour when you add an acid or an alkali to it. So let's have a look and just see what happens. So let's create our solution first that we can then test. So watch me carefully, and this is how you do this. So first of all, we're going to create our filter funnel, okay, filter paper. So what you do is you fold this in half. So fold your filter paper in half like this. So give that, so fold that in half like that. Everybody okay? And then fold that in half again. So fold that in half again, like so. So fold it in half like that. So you have like a little cone, a little tiny cone. And what you do is you stick your little finger into the cone and hold it like that. So hopefully you've got a little thing like that, and then you stick that into your filter funnel. Okay, so that's our filter and filter funnel, okay? And you just prop it in like that. Is that okay? So you're making a funnel. So this is a filter funnel. Now what I'm going to do then is going to filter my cabbage solutions. Just show you how you do that. So you pop that, move that out of there like so, and then give that a little bit of a stir, and then pour your liquid into your filter funnel, and you should have something that looks like that. And I shall just show you that on the screen. Hang on. So what you've got is something that looks like that. Is that okay? Everybody okay with that one? So this is going to come reasonably slowly, so it takes a couple of minutes to get sufficient amount of the filtrate through. Okay, so have a little practice. So I think everybody's sort of busy, so I'm assuming everybody's okay. So I'm going to put a little bit more of my cabbage juice into my filter funnel. So what that's basically doing, is filtering out any of the lumpy or bumpy bits of your cabbage, your big lumps of cabbage, and creating quite a nice, clear, purple indicator solution. So this solution here, the filtrate, is full of, absolutely ram-packed full of the anthocyanins, which you're going to use as an indicator. Okay, so just while that's filtering a little bit, I'm going to come back to our indicator paper, okay? So our indicator paper, this stuff, okay? Sometimes known as litmus paper as well, you hear that phrase. This is actually yellowy in color, and what it's basically combines a combination of different indicators, a whole different combination. Because if we look back to acids and alkalis, of course, if you might remember that we measure them on what's called the pH scale. Now, the pH scale has a numerical value, goes from 0 to 14, and it also has a color scale. It goes from deep red, which is really super low pH, very acidic, lots of hydrogen ions, right up to a very high pH of 14, and deep purple, and very low hydrogen ions, but very high OH ions or hydroxide ions, okay? So I think I've got enough filtrate. So that's what the pH paper. Now this is a very accurate one. So what we'll be able to do is actually compare the indicator that we got with our anthocyanins that we found in plants to this one and just see what the color looks like. Okay, so without much further ado, I think I've got most of my filtrate. So go back to this one. I've got quite a lot of my solution here. And I shall just pop that into there like so. Okay, so here I've managed to make probably about sort of 20 mils of my indicator, anthocyanin indicator solution from my red cabbage. Okay, is everybody okay? Is everybody okay? Can I just get a couple of okays to see that I'm going the right sort of speed and that you're not getting left behind? Got an okay, got an okay, got an okay. You guys are awesome. Fantastic. Right, next, what you need is, I'll just flip this back here. You need three of your three little tiny test tubes. So 3 of these little test tubes, and you pop those into your test tube stand like so. So so here. So 1, 2, 3. So pop those into your little test tube stand like so. Right. Now what we're going to do is going to put about 3 mils. Now you'll notice that your little pipette is what's called a 3 ml pipette. So if you squeeze the pipette and draw up 3 mils of solution, and you want to put 3 mils of that solution into each one of your little test tubes, okay? So if you do that now, one of those, one of those, one of these. Now I only put 1 mil each actually in each one of those, so I'm just going to top that one up. Okay, cool. Right, so what you should have now are 3 little tiny vials of purple anthocyanin rich indicator solution. Right, now we need to get our spectacles on. Now if you've got some gloves, you've got some gloves as well, so use some gloves. We pop these on now before we go into the chemicals. A bit of cabbage juice, not worried. The next 2 chemicals, they're perfectly fine, but always best to have gloves on, always best to have your glasses on. So if we get our gloves on, and some of you guys should have changed red and pink. Not yet, not yet. Nothing should have changed just yet, because we haven't added any chemicals to it just yet. So if we just pop our little gloves on and pop our glasses on, hang on, give me 3 seconds, 3 seconds. Okay, cool. Gloves on, our glasses. Ta-da! Now we look the part, now we're safe, let's crack on. So what we need now is the citric acid, so citric acid and the sodium carbonate. So citric acid is obviously acidic, so I said very low pH, so down to 0, and then the sodium carbonate will provide for us an alkaline solution, okay? So what we need to do is get your little pin, open up the citric acid. So open up the citric acid bottle and puncture the safety valve on this one. So all you do is puncture the safety, push that down into the citric acid, okay? And what we want now, you want 5 drops, so 5 drops only, 5 drops into one of the containers. So 5 drops into one of the containers of your anthocyanin solution. So 1, 2, 3, 4, 5, okay. Put the lid back on, this one. And then just give it a little tiny shake. In fact, if you pop the lid back on, you can give it a proper shake, like so. Now you should have got a lovely change of color. So it went from a sort of purpley color down to this nice pink, reddy color, okay? Leave the middle one, so you're going to leave the middle one, that sort of purple color, so that gives you the indicator, okay? And then with the sodium carbonate one, of course, you then pick up that pinch, puncture that little safety seal on your bottle, and again, put 5. Can you please repeat what you put in the test tubes? Yes, sure. So just to repeat, guys, in this test tube here, I've just got the red cabbage indicator, and I put the sodium carbonate. So this is the sodium carbonate. Okay, so this is the alkaline solution. I put 5 drops of that into my indicator. 1, 2, 3, 4, 5. And put that back in and go. And then put the lid back on that one. And give that a little shake. And now you see it's gone from purple, the color of my gloves, to the solution. Remember, that was the original color of the solution, to green. Isn't that beautiful? So beautiful transition of that green. And then in this one, of course, I have the acid. So I had 3 mils of the, hang on, where am I going? So 3 mil of the pipette, so there's a 3 mil pipette. If you look at the pipette, it's got 3 mils. Also, you'll notice on the side of your little tiny test tubes, you've got 3 mil measures on there as well. So 3 mils of the cabbage solution, and then 5 drops of citric acid and 5 drops of sodium carbonate. And as you can see, there's a beautiful color change in the anthocyanin cabbage solution. Isn't that cool? That's so nice. So I'm not finished just yet though. That's just one indicator. So just the anthocyanins in the cabbage give us that color change. But actually, does that really tell us exactly how much acid or how acidic something is or how alkaline something is? Well, let's just test the citric acid again and the sodium carbonate again with the litmus paper, and let's just see the color changes, okay? So for this one, if you just hold that bit of paper up, so this is a little bit of indicator paper, okay? If you put 1 drop of citric acid on one end and 1 drop of sodium carbonate on the other end, and let's just see how their colors compare with the indicator that we got. Is that okay? Can I get a couple of okays just to see where you guys are going? Is everything okay? Kind of. Okay, I've got okay. Thanks, Jake. Thank you, Manya. Thank you. Super. Right. In which case, let's splash 1 drop of the citric acid on one end. And what I'll do is I'll just change the camera. Okay, a little bit down here, push that to the side. Just going to put 1 droplet of the citric acid on one end of the paper. And then I'm going to put 1 drop of the sodium carbonate at the other end of my paper. I make it like a little unintentional litmus flag. So I just fix that up. Hang on. Let's just pick that up again. So now I created a little litmus flag, kind of close to a French flag, that one. But can you see how the color differences look here? So I've got this, the pink one's not too far off the pink, is it? I don't think the pink one's too far off the pink, but the purple one here, see this one here, deeply changed. That's a purple color as opposed to sort of green color. So this is, thank you, Jake. We're improving. So here you can obviously see the color changes are very different. So this litmus paper represents the full range of colors that you would find, and that can be found on the back of your little card. You can see how the full range of indicators can be seen and compare that with your indicator. Is that okay? Is everybody okay? Come in, Farid, yep. So I can see, oh, yes, okay. There's a couple of other questions there, but I didn't quite catch them. Yep, cool, great. Then we'll scoot on. Right. Just put that to one side and come back. Okay, so let's just sort of quickly go back a little bit to plants. So plants produce, basically, you realize that plants produce, they basically use sunlight, and they use sunlight to undergo photosynthesis. And in photosynthesis, of course, they produce the basic substances, basically glucose. So you're basically reacting carbon dioxide with water, using sunlight to produce glucose and also oxygen. So that's the photosynthetic process. And with this molecule here, glucose, where are we? Glucose. So with this molecule here, glucose, you then produce, or the plant produces, all the other chemicals that make up the plants, all the proteins, all the anthocyanins, for example, and citric acid. And now what we're going to do is we're going to have a little look at some starch. So here, look, can you see that one of the most important things, now has anybody bitten into a potato before? Has anybody bitten into a potato? Nobody? Yep. It's a bit dry, isn't it? So it's a bit dry without you cooking it, okay? Raw, give it a try. Won't kill you. It's very dry because it's full of starch grains, and of course, the first person to analyze starch grains and actually see starch for the first time was the Dutch microscopist. He first invented the microscope called von Leeuwenhoek. And he was the first person to see these funny little grains that comes out of potatoes. And if you don't believe me, chop one up, stick it into some solution, and have a look at the grains. And so what the plant does is it makes glucose, and it bonds glucose to glucose to glucose to make long chains of a molecule, and that's what starch is. And of course, the plant then uses starch to produce all sorts of other chemicals that make up the plant, okay, like lignin and cellulose and so on and so forth. Okay, and so one of the things that it also produces is ascorbic acid. So this is ascorbic acid. This is a molecule, ascorbic acid, and this prevents a disease called scurvy, which we're going to come on to. And if you want to see it as a little molymod here, that's the little molymod. So that's the molecule ascorbic acid. And believe it or not, if you didn't have that in your body, you'd die. And human beings are about the only animal that doesn't produce its own ascorbic acid, vitamin C. Lots of other animals produce their own vitamin C. all plants produce ascorbic acid, but we don't. So we have to eat ascorbic acid. It's really important, really, really super important molecule there. Okay, so we'll come back to that in a minute. What I want to do is I want to set up the next 2 experiments and then come back and explain them once they're going. Is that okay? Right, everybody okay so far? Everybody okay so far? Okay, okay, cool. Yes, we're good. Right, so without further ado, I'm going to set up the next 2 experiments and then we're going to come back and look at the effects, okay, because it takes a couple of minutes for them to come back and look at them. So what you need now is your potato and your lemon, okay? So we're going to cut a slice out of the potato and a slice out of the lemon. And in this experiment that we're setting up, we're looking for starch. So one of the evidence that a plant is actually making starch. So if I bring it back to the table and I bring my potato and my lemon, what I'd like you to do is cut 2 slices of each. Okay, so if you cut 2 slices of each one, so cut 2 slices of potato and 2 slices of lemon. So you do that now, reasonably small, so about sort of 2-3 centimeters sort of thick. Okay, so I'm going to just chop the edge off this one. So that's 1 slice here, and then another slice. Save the rest for dinner. I'm kidding. Don't eat any of these things. Once you've played with these ones, these go in the bin or in the recycling thing. Okay, so 2 bits of potato and we want 2 slices of lemon. Okay, so 2 slices of lemon. Right, cool. Put those to one side. Now, with one of the slices of lemon, cut it into 4, and with the other slice of potato, cut that one into 4. But before you cut one with the same knife, give that knife a wash with the water I asked you to get in the first place. So what you're going to do is get one of your little cups, in fact 2 of your little cups. Voila, voila. You're going to put 4 little slices of potato in here and 4 little slices of lemon in here, but wash your knife in between. Is that okay? So 4 little bits of potato in here and 4 little bits of lemon in here and wash your knife in between. So watch me and I will show you how this is done. So here with our lemon, cut that into 4 and pop that into there like so. Then take your little knife and give it a good wash, a rinse. Now take another bit of potato and cut this one into 4. And stick that into there like so. Whoops. And give that a little rinse. Okay. Now I think we're all good. Now, can they use orange instead? Yes. If you haven't got lemon, you can definitely use orange, or you could use red pepper, or you could use lime, or you could use le banane. You could use banana. Anyway, so right, is that okay? Yep, is everybody okay with that one? We're okay, we're okay. Get the impression we're all okay. Fantastic. Right, now what I want you to do is put boiling water from your flask, so boiling water, thanks Jonah, thanks Becky, put them into your cups. I'm going to leave those to one side. So put some boiling water into your cups and then pop those off to one side. Fantastic. So pop that lot off over here and leave that set. So we need to leave those cooking, as it were, for about 5 minutes. The next thing we need to do then is set up our next experiment. And in order to do that, we're going to in this experiment, we're going to look at citric acid. So the one we just set up is for starch, one of the products of photosynthesis, or eventual products of photosynthesis. And now we're going to then set up and look for ascorbic acid, which, by the way, all plants need to help. And photosynthesis is actually quite a damaging process in part. And all the roles of ascorbic acid in plants actually aren't well known. But we do know now that ascorbic acid in plants helps protect the plant against some of the damaging products of photosynthesis. So it's very useful to help protect the plant. And of course, we need it because, of course, vitamin C, ascorbic acid, is essential for making all our soft tissue or soft matter in our bodies. Does anybody know what the disease is? I gave you a little hint earlier. Does anybody know what the disease is that you get if you don't have any vitamin C? Anybody? What is the disease you get? If you scurvy, A-law, fantastic. Yeah, it's scurvy. And scurvy, thankfully, you hardly hear about today, but it does. Thank you, Jake. Fantastic. Yeah, it's scurvy. It's a horrible disease. If you don't have vitamin C, and you must never do this, don't deliberately have no vitamin C. I think that sounds right English-wise. Don't never have any. Who knows? But if you don't have vitamin C, what did you do after you put in the cap? Which cap? Naya, which cap? Booster scurvy. Yes, indeed, Becky. It's a horrible disease. First of all, it makes you feel really weak to start with. That's the easy set. You get very, very, very weak. You get headaches. Your gums start to bleed. Your teeth fall out. You start to bleed from your eyes, and then you die. It's just not a nice disease to have, and it's very painful all the way down. And actually, this is one of the reasons that, actually, I'll come back to that. Let's set up the next experiment. Okay, I'll come back to that. Okay, what I'd like you to do now is we're going to do the make up an iodine solution. So in order to make up the iodine solution, what we need is this one here. Sorry, just flip that back. So this is the iodine. So we're going to do this one. We're going to look for ascorbic acid with this one, okay? And we're going to use iodine. So we've got to create the iodine. So in order to create the iodine, we need the following chemicals. You need, and glasses back on, you need some potassium iodide solution, which has the iodine, and you have some copper sulfate. And when you react the copper sulfate with the potassium iodide, the reaction, actually quite a complex series of reactions that goes on here, but one of the products is iodine. There's also copper iodide and potassium sulfate. But these are the two chemicals we need, okay? So, without much further ado, if you get one of your little cups, so we need one little cup here, just come back to screen. So one little cup, this one, we're going to react our reagents here. Okay, so one little cup, and with our two potassium iodide and copper sulfate, you also need one of your spoons. So I'll just check everybody's okay with this. So you need your copper sulfate, you need your potassium iodide, a spoon, and a cup. Okay, you also need your little pad, your little absorbent pad, and you also need your sheet of paper. Is that okay? And if I get a couple of okays, I think everybody's okay with me. So sheet of paper, yes, of course, Jonah, yes, of course. Sheet of paper, cup, potassium iodide, copper sulfate, and absorbent pad. And you'll also need, of course, your tray, the little say tray. Is that okay? Right. So without much further ado, what you do is the following. So in your cup, first things first, is put the copper sulfate. So you want one big spoonful of the copper sulfate. So one big spoon of copper sulfate in here. Okay. Then you need two spoons of the potassium iodide. So 2 spoons of the potassium iodide into there like so. 2 nice generous spoons of potassium iodide. Stick that there like that. And then give that a swirl around. So just give that a nice big swirl around for about 10 seconds, okay, about 10 seconds. And you should see a nice blue, sort of brown, sludgy mix there. So actually in here, you've got potassium sulfate, you've got copper iodide now, and you've got free iodine. You've got free iodine. It's the iodine we're interested in particular here. Just give that a nice little sludge around. Now, what you then do is get your absorbent pad. I'm just going to give that a little bit of a push down and soak up all of that solution there. So give that a good old soak up, about 5 seconds, 2, 3, 4, 5. So that's soaked up most of my iodine solution here. Now what we're going to do is we're going to paint this, and I shall show you how to do that with the other camera, okay? Just move the camera around. Okay, so what you do is with that end, going to paint this paper, so with one layer only. Like so. Oops. To get quite a nice covering, you might have to give it a little squeeze. Okay, that's cool. Stick that back in there. And then what you need to do is place the potato and the lemon onto the sheet like that. And just press it down a little bit. So press it down. So press the lemon down and then press the potato down on the sheet like that. Okay, just going to get a little bit of tissue paper, rub that down. Okay. Okay, I think that might have taken some people a little bit of time to do. Just throw that away like that. So at this point, now we need to leave that to one side so that reaction can take place. Ellen, excellent, fantastic, cool. So I said, just for those who are interested in the reaction, the reaction, of course, as you had copper sulfate here, and it's reacting with potassium iodide, and that produces copper iodide, potassium sulfate, and iodine. It's the iodine we're particularly interested in this particular case, because what actually happens in this reaction is the ascorbic acid is quite happy to give away hydrogen ions. It's quite happy to give electrons effectively away. And what happens is the iodine that's there is very happy to take them, okay? And in going from an iodine a molecule effectively to a charged ion, it changes the color. So you can see when you put it onto the paper that the iodine stained it brown, that's the iodine, okay? And if it grabs some electrons, it goes into iodine ions, and that goes from brown to colorless. So the theory is here, if we're looking at comparing different substances that plants produce, the amount of ascorbic acid, vitamin C, that plants produce, now we've kind of got an inkling. Have you guys, you must have bitten into a lemon, haven't you? Who's bitten into a lemon before? Neat, yes, indeed. Nope. Well, if you haven't tried, you must try soon. Very good for you. A bit tart. It's very acidic, so it's full of ascorbic acid and citric acid and a number of other acids, in fact. And so it's quite sharp. So there's a probably good chance that the lemon has a bit more vitamin C than the potato. Does anybody think that do potatoes have vitamin C, do you think? Do potatoes have vitamin C? Katie, Elor, Jake, yes, Ellen, yes, they do. Fantastic. Yeah, actually, you guys did. They do. Potatoes do have vitamin C in them, and they don't have very much. Probably about 19 milligrams per 100 grams, something like that. It's about sort of 19 to 20 milligrams per 100 grams of vitamin C. But interestingly, do you know that if you boil the potato hard, you get rid of all the vitamin C. It's a very unstable kind of molecule. So if it's exposed to light, it disintegrates. If it's exposed to heat, it disintegrates pretty well. And that's why a lot of people, when they take Lemsip, when they got a bit of a cold, I think they think the vitamin C is going to do them good. Well, a couple of years back, the companies took the vitamin C out of the packet because they were killing it with the boiling water. There was no point in putting the stuff in the first place. So anyway, all by the way. But a really interesting aside from this one is the fact here, if I could just introduce you a little bit of history, you may know that the British, back in the 18th century, the British were called limeys because of this guy here, Captain James Lind. And he figured out, he's a fantastic scientist. You guys really encourage you to go and research about him. Really interesting doctor, fascinating scientist. And he basically realized that if this scurvy disease that all the sailors were getting, if they had limes and oranges and potatoes, fresh fruit, they didn't get scurvy, and therefore they could sail happily on the seas and go and fight properly. So he basically realized that when the British Navy went to sea, they packed the hull full of limes and lemons and stuff like that, and therefore they were able to fight. So that's why the British were called limeys. Really interesting doctor, really interesting scientist. Please do read up on him, James Lind, that is. Yeah, it's an amazing, lovely little bit of history. He did so much other stuff as well. Little joke for you. Anybody up for a little joke? Corona, not Corona. No, Corona isn't affected by it. Hello, yay! Okay, listen up. I keep shouting, broccoli, cauliflower. I think I might have got a case of florets. Hey, get it? I keep shouting, broccoli, cauliflower. I might have got a case of florets. Anyway, just slow. Oh, come on, Jake, that's not very good. Thank you. Okay. I've got more jokes. Right. So we need to get back to experiment number 2, where we're going to look at starch. So the chemistry for this one. Oh, sorry, Alan. Okay, Alan, just for you. Why did Henry VIII's wife have little bite marks all over her? Why did Henry VIII's wife have little bite marks over her? No, not quite. Because he chewed her. Get it? Because he chewed her. You gotta like that one. Oh, right, okay. A bit, okay. Right, let's move on to the last of the DIYs. So what we need to do here is we need to test our warm lemon solution and our warm potato solution. So in order to do this, you need this particular card here. So is this food starchy? So we're looking at the starchy one here. Okay, so you need this particular card, and here is the equipment. This is the method I'm going to be following. Okay, so in order to do that, you need your little, let's move that to one side and move that down here. So you need one of those, so your little tiny test tube. You need your potassium iodide again, so potassium iodide. You need your copper sulfate again. You will also need your pipette, so a little bit of pipette. And you also need your spoon. So this is your spoon. Okay, so in order to do this, you're going to put, one big teaspoon of copper sulfate. Thank you, Farida. Hey, can't please everybody. I got one more joke for you then. Okay, so you want 1 teaspoon, 1 large teaspoon of the copper sulfate. So 1 large teaspoon of the copper sulfate, and pop that into your little test tube. So 1 large teaspoon of your copper sulfate into the test tube. So that's 1 of the copper sulfate. Where are we? Here we are, copper sulfate. Yes, Katie, definitely. There's one more joke to come. I think I've got time for one more joke. Okay, now we need two large teaspoons of the potassium iodide. So 2 of those, 1 and 2. So 1 and 2 of those. Then put the lid back on this one, screw it down tight. Mania, carbon dioxide contains enough carbon to make 9,000 pencils. It does. It depends on the size of your pencil though, Mania. Okay, and the amount of graphite you need to put in your pencil. So give that a nice little shake for about 5 seconds, something of that nature. Right, so put that back to one side. Now you need to get back your hot lemon solution and your hot potato solution. So hot lemon solution and hot potato solution. So if you get those back, but before you do that, one of the interesting tests for starch is if you add iodine to starch, the reaction produces a black chemical. The interface with starch and iodine, the iodo-starch compound, produces a black substance, and it's clearly very black. So if one of these solutions has got starch in it, when we put the iodine solution, which is brown, it should go really, really black, okay? So let's just try that now. If we scan back to the table again, and what we want to do is put about 5 drops each. So we're going to put 5 drops, so that's 5 little drops into the 1, 2, 3, 4, 5, and then 1, 2, 3, 4, 5 into there. I'll just stick that there like so. I'm going to give that a little stir. Give that a little mix, and then give that a little stir, and then just show you what this looks like on camera. Now, can you see clearly that this one, the potato one, has gone really, the potato one rather, has gone really, really, really dark black. So this one is full of starch. So potatoes are full of starch. So the potato plant manufacturing glucose through photosynthesis, manufactures lots of starch, and then sticks that down into the tuber for use later on during the plant cycle. And here, did you see the lemon has gone hardly black at all, but there is a little tiny bit of black in here. Can anybody tell me what might have happened? Why there's a little bit of black in there? Anybody? Anybody being particularly observant? Hello? You've all gone quiet. You've all run off. Anybody? Everybody okay? Because of potassium? Not quite, Mania. Not quite. That's a nice suggestion. Use the same spoon. Christy, yes. Farida, it did. And this is a nice little example of cross-contamination in chemistry, okay? So actually, I dropped the potato onto the lemon a little bit. I wonder if anybody noticed that. And of course, it's got slightly contaminated, so that's not the result I would expect. Because actually, there is 0 starch in lemon, not a scratch in lemon, okay? It's basically all either sugar, ascorbic acid, a bit of water, and various other stuff. Is that okay? Cool. So we basically got a starch test, and we produced a load of high starch reaction with the iodine in the potato and none in the lemon. And we've done a little bit of look at anthocyanins, and we've seen the wonderful ways in which they change color with our citric acid and our sodium carbonate. And then finally, last but not least, let's have a little look and see what our lemon and our potato has done on our sheet. Is that okay? Okay, right. So if I just do the great reveal, if I pull the lemon off, oh, look at that, magic. And then pull the potato off, awesome. So can you see that the lemon has got lots and lots of missing iodine. Well, it's actually missing, but it's changed into iodine ions, which of course are colorless. And here the potatoes produce a little tiny bit, pardon me, of discoloration here, which is in keeping with the fact that it's got a lot less. Don't think it's better on that one. Let me see that probably a little bit better. So this one is obviously the lemon with a lot of discoloration here because there's lots of ascorbic acid, and this one's got a little bit because it's got a little bit of vitamin C in it. Is that okay? Little tiny whirlwind tour of plant chemistry. I hope that was okay. I hope that was useful. I'm very happy to field any questions. You've got to field any questions. Katie, if it didn't work, try it again. Try it again. Alan, I'm really pleased. Great, excellent. You're very welcome. No questions. Manya, no questions. Jonah, everything went well. Lovely to hear it. Jonah, thank you very much. You're very welcome. I'm back next week. And next week, the schedule is slightly changing, I think. We've got 3 separate sessions during the course of a Wednesday and a Thursday. I'm just happy that you didn't want. Excellent. You're very welcome. Aaron, you're very welcome. Anyway, lovely to have you guys on board. I'm so delighted to be back with you, sharing a little bit of fun and a little bit of chemistry. So all I've got to say for you guys is stay well, stay safe, stay away from COVID, keep experimenting, keep inquiring, keep messing, keep your glasses on. I shall see you next week. Thanks ever so much, guys. I love and leave you. Goodbye. Hey, but still no questions. Okay.